Okay, binding of iodine with the thyroglobulin hormone is known as organification. This is the step here. The iodine binds with the thyroglobulin protein is known as organification, and this organification process, this binding of iodine with the thyroglobulin protein is known as organification. Although if peroxidase enzymes are if not present, iodine gets bind with the thyroglobulin, but it is a very slow process, so it requires the enzymes peroxidases, and peroxidases helps the hydrogen peroxide, and this combination of nascent iodine with the thyroglobulin protein boosts or it increases or it goes on it goes speedily. This is the iodination process of the tyrosine residue. Iodine gets iodine binds with the tyrosine residue, and this then at this position when iodine gets attached, this is known as monoiodotyrosine, and then it occur in the presence of the enzyme known as iodinase, and then this monoiodotyrosine gets bind with another iodine atom. It is known as diiodotyrosine. When these monoiodotyrosine and the diiodotyrosine gets bind with each other, this is the formation of that 3,5,3 prime. Okay, then this is the diiodotyrosine, 3,5,3 prime diiodotyrosine. Then this is converted into the thyroxine T4. This is the T3. It has the three. This is the T3. It has 3 iodine molecules, and this is the T4. It has 4 iodine atoms. Storage of thyroglobulin. The thyroid gland has unusual ability to store large amount of the hormone, and after the synthesis of the thyroid hormone has run its course, the thyroglobulin molecule contains 30 molecules of thyroxine molecule and triiodothyronine molecule. In this form, the thyroid hormones are stored in the follicles, and if there will be some deficiency in the body or some defect in the thyroid gland, approximately you can't detect the problem in the thyroid gland for up to 2 to 3 months, because thyroid hormones are already stored in the follicles are enough for the functioning of 2 to 3 months. Okay, release of thyroxine and triiodothyronine from the thyroid gland. Thyroglobulin itself, it is not released into the circulating blood. Instead, thyroxine and triiodothyronine, both these hormones are first cleaved from the thyroglobulin molecule, and then these free hormones are released. This process occurs as follows. The apical surface of the thyroid cell sends pseudopodia, as I already told you with the help of diagram. These pseudopodia forms extension, and then these encircle the T3 and T4 to form the pinocytic vesicles. These pinocytic vesicles then these pinocytic vesicles has a fate to combine with the lysosomes, and then these lysosomes are responsible for the digestion of the vesicle containing the enzymes, and multiple proteases among the enzymes digest the thyroglobulin molecule T3 and T4 and are released, and then these diffuse into the blood, and remaining substances are recycled. Okay, 93% of the hormone released is in the form of T3, and 7% of the hormone is T4. 93% of the thyroid hormone is T4, and 7% of the thyroid hormone is T3. Okay, now T3 and T4, how they are transported to the all body cells, because all of you people know that T3 and T4 are responsible for increasing the basal metabolic rate of the body. Whenever these hormones enter into the blood, these hormones has high affinity with the plasma proteins present in the blood, and these plasma proteins are synthesized by the liver. They mainly combine with the two type of proteins, thyroxine binding globulins with, and much less so with the thyroxine binding prealbumin and the albumin. Because of the high affinity of the plasma binding protein for the thyroid hormone, so it is so thyroid hormones, thyroxines is released slowly on entering the tissue cells. Both thyroxine and triiodothyronine again bind with the intracellular proteins, and the thyroxine binding more strongly than the triiodothyronine, so they again are stored in the cells, but this time these hormones are released on the requirement from the cells slowly. Okay, next topic is the physiological function of the thyroid hormone. Most of the T4 is converted into T3 by, because cell has receptor more for the T3 because most of the cells has receptors for the T3 instead of T4. Okay, now a thyroid hormone activate nuclear receptor mechanism. This will be explained with this diagram. Here you can see T4. If thyroid hormone enter in the form of T4, it is necessary to convert it into T3, and this occur with the help of hormone known as enzyme iodinase. If T3 enter directly, then it enter into the nucleus. Okay, nucleus has a DNA, and a specific gene of the DNA has receptor sites for thyroid hormone. Thyroid hormone has a dimer. Thyroid hormone receptor is a dimer. It contains two units: retinoid X receptor and the thyroid hormone receptor. Then it binds with a specific portion on the gene on the DNA, that is known as thyroid hormone response. Okay, these hormones easily enter the cell membrane because they are these hormones enter with the help of specific receptor proteins present in the cell membrane, because they are protein in nature. Then when thyroid hormone gets bind with the receptors, it stimulates the transcription, and the resultant product of the transcription is the messenger RNA. Then the translation of this messenger RNA occurs, and the resultant product is the proteins, or this process is known as translation. So these proteins are involved in the metabolism. It enhances the mitochondrial rate, sodium, potassium, ATPase, oxygen consumption, glucose absorption, gluconeogenesis, glyconeogenesis, lipolysis, protein synthesis, BMR. BMR is the basal metabolic rate. All these processes are increased. So what is the effect of these proteins on the cardiovascular system? It increases cardiac output, tissue blood flow, heart rate, heart strength, and respiration. These proteins are also involved in these hormones are responsible for increasing the growth of the body cells, and CNS development is also involved, and many other systems are also